So yeah, as Dan wrote, the environment has effects both between generations, natural selection, the environment favoring some traits at the expense of others. But the environment can also have an effect over the course of development in shaping the phenotype of an individual. So it's not the natural selection part of the environment, it's the how does the environment affect traits along with genes? They do. Um, so Galton founds and coins the field of eugenics, which means, it sounds like it means good genes, and it kind of does, but it's well born. Actually, both genes and genics come from the same Genesis root. Um, but there you go. And I mean, he wrote some goofy stuff. So he wrote a book. He produced a beauty map of the British Isles, to sort of highlight what areas had the most attractive people. Um, Galton is in some ways um, uh, less damaging than Pearson and Fisher later on, and sort of Davenport in the US, and obviously the National Socialists in Germany, in that you can think of negative eugenics and positive eugenics. Negative eugenics is restricting reproduction of groups, and that could be done by sterilizing or, you know, or 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 worse. Um, Positive eugenics is where you encourage the best and the brightest to have more children, or you figure out how to make sure that the very best and brightest only mate with each other. Um, so Galton was not recommending things like sterilization, partly because the technology for sterilization was grisly at Galton's time. Right later on, um, vasectomy and salpingectomy, much less invasive surgeries that are not awful, um, were developed, and that actually probably affected the scope of what eugenicists thought we should be doing. Um, so Galton wasn't that different from Darwin or Haeckel, other early evolutionists, who thought that there was a hierarchy of races. Um, However, um, Galton thought that, natural selection was going to sort of take care of less fit races. So Galton was mostly concerned about within Britain, are we going to, maintain our crop of geniuses every generation? We could only do that through positive eugenics. Um, but that's not where eugenics ends, it's just starting. So interestingly, there was a famous genealogical study by R.L. Dugdale of a family that the title is The Jukes, A Study in Crime, Pauperism, Disease, and Heredity. That was published in 1877. So this is an American, I guess, sociologist. Um, and interestingly, Dugdale did the study not to make arguments about eugenics or, heritability um, of of traits, but actually just to say that, like, there's a cycle of poverty that we need to break through environmental interventions. Developing a public school system, which was still very new in the 19th century. Um, but the study described, two or three generations of a family that was basically always in trouble. Um, so I want to get to we're gonna get from that to eugenics in the 20th century. Um, the other thing to know about, like, eugenics is that it's kind of a reflection of economic theories that celebrate individual achievement and social Darwinism, competition, leading to, positive outcomes. So eugenics is becoming more popular, partly because Mendel's discoveries are becoming well known in the 20th century. So the understanding that genes can affect any traits is kind of on people's minds. And so there's Galton's estate endowed a chair of eugenics that was held by Pearson and then later Ronald Fisher in 1933. And then we see in the United States, you see eugenics is becoming more popular in county fairs. You'd get these things called fitter families competitions. So just like people are showing their best livestock and best produce and how their breeding plan, designs are, increasing the genetic quality of animals and food plants, families could enter themselves as, having good genes. And then we're going to start to see in the 20th century in the US, it starts in the US, restrictions on reproduction among those who are deemed to have bad genes. So this is a eugenic display from a state fair. And you see, over here, there's genetics of fur color in, showing sort of Mendelian inheritance in, I think, in rats. And then you have, anyway, and then you have other displays from the American Eugenics Society. And then you have these fitter family competitions. So this is a family that won. Surprisingly, they're they're white. Oddly enough, they're mostly wearing glasses. But anyway, they they're what we should all be aspiring to, or something. So the novel development in the U.S. is actually the negative eugenics, the restrictions on marriage, and then later forced sterilization. So there were rules and laws put into effect. By 1914, over half of states restricted marriages by people with mental defects. The other thing that's happening in the early 20th century is the development of IQ testing, right? So and the funny thing about IQ testing is it's happening. So IQ testing is just starting. So I'm not going to be able to get into it here, but the history of IQ testing is fascinating because the huge experimental data set was the application and giving IQ tests to all these young men being drafted into the army to fight in World War I. So that's 1914. And the interesting thing is there were, all that testing was done, and it was startling, recent immigrants and groups from different ethnic groups, races that you would not think of as races, like Irish and Italians were seen as different races then, would score low on the IQ tests, and this was seen as both validation of the testing and strong evidence of the genetic differences among races. And it wasn't until a couple decades later where these generations of recent immigrants um, uh, adjusted to American society and were successful in all walks of life, that the sort of predictive ability of IQ tests was strongly questioned. But like in the early 20th century, you had this new testing and ability to define people. So I don't remember what the things were, IQ of, less than 70 or something, in some states would not be allowed to marry. But this was seen as insufficient because then, as now, people don't have to be married to have children. That's beyond the scope of this course. So Davenport, who was the founder of Cold Spring Harbor, which is still, an incredibly prestigious genetic lab, but it used to be a eugenics lab, um, was arguing that we shouldn't just have restrictions on marriage. We're going to have to do compulsory segregation of defectives. That means putting them in institutions, and possibly sterilizing them. So, as I just said in the previous slide, because I always say the wrong thing with the wrong slide, the Simon Binet intelligence tests are just being developed then. And so there was um, a significant increase in asylums and hospitals that were already present, but now they were being expected to hold a larger portion of society, not just the really ill, but just the people who didn't score high enough on IQ tests. And so the hilarious thing about this was that it was expected that needing all these asylums and hospitals was just going to be a temporary cost. And as the bad genes were pruned from society, there'd be more income from taxes because you'd only have, smarter people in the workforce, and they'd be able to close down the institutions. But it turns out that housing people in institutions very expensive, and of course had no effect on the proportion of people who did not do well on IQ tests, because it was um, you know based on a culturally dependent test. The other thing is that sterilization was much less invasive now. So now there were less invasive. So it was both more like possible and plot and uh, you know politically more acceptable to have sterilizations. And so people were sterilized, sometimes on the basis of IQ tests. The first person to be sterilized because they were, declared feeble-minded was Carrie Buck, and her sterilization was was challenged in court, went up to the Supreme Court, where it was upheld. So this is Oliver Wendell Holmes, was the Supreme, Chief Justice of the Supreme Court at the time, um, and described her as feeble-minded white woman who's committed to the state colony, an asylum, an institution. She is the daughter of a feeble-minded mother in the same institution, and the mother of an illegitimate feeble-minded child. Um, And in his argument, he's saying, in his decision, this is an excerpt from this decision, 1927, it is better for all the world if, instead of waiting to execute degenerate offspring for crime, or let them starve for their imbecility, society can prevent those who are manifestly unfit from continuing their kind. The principle that sustains compulsory vaccination is broad enough to cover cutting the fallopian tubes. 3 generations of imbeciles are enough. It's statements like this that give vaccination a bad name, which is incredibly frustrating. But it's also really worth learning about Carrie Buck's case in some detail. So I would recommend a really short article that I posted to Blackboard. It's an essay by Stephen Jay Gould, um, who went back and did the research to find um, Carrie Buck's, elementary school report cards, grades, and those of her daughters. And she was um, there's lots of different ways to define, um, I guess, to define intelligence or to define, severe developmental disability. And that was not her. She was fine. She was a B student in elementary school. What happened to Carrie Buck happened to many young women, which was that she got pregnant young before she was married. And one of the ways that families dealt with children who got pregnant, 17, 18-year-olds, I don't remember how old she was, um, is 16, is that they would put them in mental institutions because that's where they could have the baby outside of society and then people wouldn't see them and they wouldn't be embarrassed, the parents. And so Carrie Buck was institutionalized not because she was mentally impaired, but because she had a child out of wedlock. And because of that, um, she and her daughter were sterilized. And it's a pretty shameful legacy. Um, I think we'll have it in the slide, in the next slide. About 70,000 was what I remember. 65,000 women were sterilized in the U.S. Ending in 1981. Was the last person sterilized because under these laws. Um, and yeah, it is pretty late. Um, so what led to the ending of this policy was some of that IQ testing of immigrants during World War I that I talked about. So that IQ testing was used to significantly limit immigration, which led to, many more people being killed in World War II than had to be. But it was also clearly documented and observed that the immigrant populations that did poorly on IQ tests during World War I integrated just fine into society, and the testing was, the value of that testing was clearly called into question. People, studied and learned to recognize the cultural biases in these tests. Like, one example, I don't think I have it on a slide, in the World War I version of the test, you'd be shown a house and it doesn't, and you have to recognize what's missing, and it doesn't have a chimney, and so you're supposed to recognize the house doesn't have a chimney, and that's the correct answer. And people from Italy would often write down that it was missing a cross on the door, and they'd be graded, wrong for that question. Very, strong cultural bias to the question and the answer and what you're going to notice is missing. And so there you go. And then the other thing is also, the Holocaust genocides that were done in the name of eugenics and eliminating inferior races also, gave sterilization a bad name. Yeah, so other countries did. What's interesting is that eugenics starts in England, influences a movement in the United States that leads to these sterilization laws, and then it's the rules in, about half the states in the U.S. It was a state by state sort of policy thing, are directly credited by the, you know um, whatever they call themselves, by the National Socialists, by the Nazis in Germany, to put in harsher but not qualitatively different, just quantitatively much more exuberant and enthusiastic eugenic policies. So one important question is to sort of ask, like, what is the relationship between Darwinism and eugenics and Nazism and the Holocaust, and how should that make us view sort of evolutionary theory in general. So, and I put one of the articles, I think it's not Weindling, I think it's Weichert, anyway, in a 2010 edited volume by Alexander and Numbers. I also post the article that I'm going to be using for most of the next few slides on Blackboard. So there are lots of quotes from different, high-ranking officials in the Nazi Party praising Darwin and eugenics, but it is not clear whether Darwin inspired racism or were just being called out in an effort to give racism that was already there a sort of scientific sheen, which is why it's an important question to try to understand, like, to what degree did sort of mainstream biologists in Germany, in the United States, support Nazism and different kinds of eugenics. So the German eugenics laws were modeled on California's eugenics laws. However, they were enforced much more enthusiastically. So between 1927 and 1981, 65,000 people are sterilized in the US. About 400,000 are sterilized in Germany between 1933 and 1945. So, order of magnitude higher scale. Hitler didn't have much background in biology, failed painter and veteran. But Himmler did study genetics and agriculture. The idea of a struggle for existence that Hitler writes about in Mein Kampf is earlier than Darwin. And the other thing to also think about is, like, a lot of the concepts of race and racial purity are very different for Hitler and other Nazis than Darwin. Remember, Darwin is I'm not saying Darwin was not a racist. He thought that there was sort of a hierarchy among different races, but Darwin also thought that there were no firm dividing lines between, within species or between species, between humans and animals. So Darwin is not going to be saying that there are, like, immutable genetic differences between races or even between species. The thing that's different about both the later eugenicists and the Nazis is that they're really thinking just about group level selection and heritability, right? They think of the important contests are not the contests among individuals that differ in traits and reproductive success, but the sort of world conflict between races, and that, it's a contest where in the end only one race is expected to survive, and that's different than Darwin. And so there, what you see here are trying to show how Jews have the worst traits of many degenerate races, and it's a sort of uniquely bad combination of traits from other races. So Mendelian genetics was was approved, but this is trying to summarize how mainstream scientists in Germany sort of overlapped with Nazi ideology, and the take home is that mainstream science in Germany was quite enthusiastic about Nazism, not always, but the exceptions sort of show the general trend, which is that academic departments were mostly willing and enthusiastic to purge themselves of Jews in the faculty, um, and partly because the remaining faculty then would benefit from taking over their labs and stuff. Um, Not every individual and not every department acted this way, so not everyone was willing to accept a post that was vacated because the occupant, who was Jewish, had been um, fired or shipped, imprisoned in a ghetto or a camp. Um, but others became, active members of the Nazi Party, like Konrad Lorenz, and after the war would say that they were forced to. So it's an interesting question. There you go. And some academics would flee. Not just non-Jewish academics left because they didn't want to cooperate with that program. Um, However, basically mainstream science worked together with the Nazi program. And so they would collaborate in order to basically like do things like share body parts or imprison members of families, so that with interesting genetic conditions so that they could be studied, right? So there were, horrific experiments that were carried out at the concentration camps, and those were carried out with, sort of funding in the framework of the German equivalent of our NSF, right? So if the Deutschland Federation for Genetics or whatever had a grant to study hereditary disease, you could like request individual family members from, Jewish or probably gypsy families, and then make sure that you could, like, if you're interested in heritability of cataracts and as an eye condition, you could make sure you got the eyes from everyone from the family. Um, on the bright side, um, the World War II sort of discredited genetics. Although there weren't any trials of geneticists at Nuremberg. Um, and I think I would probably, um, agree with the conclusion of a statement from the Anti-Defamation League, which where they argued that Hitler did not need Darwin to devise the final solution, and Darwin and evolutionary theory cannot explain Hitler's genocide. However, there was clearly sort of collaboration between mainstream science and the, eugenic and genetic theories that were developed in England and the United States, were, different, were not necessarily genocidal, but they definitely sort of provided support and comfort to um, um, to sort of Nazism final solution. It also had a huge impact on how animal and human behavior was studied in Europe and the U.S. So the fact that Lorenz was stayed in his academic position and collaborated with the Nazi Party kind of split the study of animal behavior in the U.S. As opposed to in Europe. So it's a really interesting thing. So the the study of animal behavior starts in Europe, and it's called ethology, and the focus is on real natural behaviors of a wide variety of organisms, sometimes in the wild or in at least environments that are trying to recreate the natural world, naturalistic environments. So you think of learning imprinting in ducks with Lawrence. You think of the digger wasps using landmarks to orient to their nests. Um, Fighting behavior in fish and learning exactly what parts of the fish, the movements versus the markings versus which markings versus the eye spots are needed in order to have contests between them. And of course, Von Frisch and the dance language of bees. So all of those are um, are sort of real natural behaviors in the wild or in normal environments, and they often focus on instinctive behaviors. So all the language of sign stimuli, um, so sign stimuli. If you learn any behavior in psych, you're going to learn these um, uh, fixed action patterns, and the innate releasing mechanisms that stimulate the fixed action patterns. So Um, so all of these are studied by ethologists. Sign stimuli is like, what is it about a stimulus that's actually going to then release the behavior? Fixed action patterns can include things like a ritualistic dance, or the most famous examples are the movements that retrieve an egg from if it's fallen out of the nest in ground nesting birds. So that was studied because it was a ritualized behavior that's kind of always done the same way. Um, there's some study of learning, but it's not associative learning tied to rewards. It's usually part of an innate program. So imprinting on learning who your parent is, using landmarks to learn how to get home, or learning from others where a food source is. That one's rewarding. The other kinds of learning, imprinting, is not associated with a reward. That's not what's driving the learning. Um, and this is studied in Europe, but the focus on instinct is now grossly offensive to many people studying behavior in the US, partly because behavior is being studied in psychology more than in biology departments. And the interest in studying behavior in animals, comparative psychology, is to try to understand behavior in humans. And there's just the personal antipathy that the thinking that Lawrence was a Nazi, and that there's a slippery slope from focusing on instincts to genetic behavior, to then thinking that human behavior is genetic, to thinking that differences among races is genetic, leading to um, leading to you know um, perhaps genocide, right? And so, as kind of a reaction to this, there's a focus on what's called general process learning. The idea that all behaviors develop through learned associations between behaviors and actions and either rewards or punishments, right? So comparative psych, in contrast, isn't focusing on a wide variety of organisms. It's pigeons and it's rats and maybe monkeys, but it's mostly rats. And there's this idea of general process learning, and actually general process learning is like one kind of learning, and it often leads to, you have to do like incredible, like um, you have to jump through crazy hoops um, to say that, to make animals that have very different like biases in how they usually learn, to get them to try to learn in the same way. end up having to try to train earthworms to push levers and stuff. Um, it's kind of weird. Um, but there's a much greater focus on learning, and especially associative learning, learning to associate behaviors with punishments or rewards, and then also more on neurological and hormonal mechanisms. Um, and there's this idea that Morgan's canon, which is kind of a version of Occam's razor, which is this idea that um, only use the simplest um, cognitive explanation, or basically deny that there's a cognitive explanation possible. Um, and there's a complete sort of de-emphasis on learning that isn't associated with rewards, which is a little bit weird because there's a lot of learning, especially a lot of human learning, that is not, at least not in a simple sense, reward based, like language learning, song learning in birds. Um, is, the offspring are not clearly being rewarded, although you could say that the attention that they get when they make certain sounds is a sufficient reward. In other words, a lot depends on how you look at it, but anyway, the general process learning can be sort of summarized by Skinner: pigeon, rat, monkey, which is which, it doesn't matter. Once you've allowed for differences in the ways in which they make contact with the environment and in the ways in which they act upon the environment, what remains of their behavior shows astonishingly similar properties. So this was very influential. Um, and what then led to a bit of a um, merging of studies of animal behavior in Europe and the United States was actually Tinbergen, both his personality and willingness to meet with comparative psychologists and develop friendships and collaborations, and then also this framework that you can study sort of adaptive behaviors and their functions, the ultimate questions, and you can also study the proximate mechanisms, learning um, how genes in the environment interact, and then neural and endocrine mechanisms of behavior. Okay, so that is super quick, um, like tour through why um, the role of genes and behavior is a sort of philosophically and historically fraught topic, and why many people are maybe sometimes too cautious, but really cautious about talking about and studying the role of sort of genes and behavior. Okay, but now I'm going to talk about heritability. So heritability, the main take home is heritability is not what you think it is, and heritability is not what proportion of a trait is controlled by genes. That's the take home, and now it's going to take me a long time to develop that take home message. So we'll do an imaginary but plausible example of running speed in dogs. So you time the dogs, you have them chase like a cat or something, or a treat, and you time them, and there's variation. Some dogs are going to be very fast, and some are going to be very slow, and that's okay. Remember too, that all dogs are the same species, so they can all interbreed, make puppies. But you'd get a distribution even if you were looking at a single breed. And the average is in this sample is 10 meters per second, and a generated data sets so that the graphs actually match the numbers. Now there's a variance, and the variance is the average of the square of the difference between each observation and the mean. So if you look at all these are, if you look at this, this is the difference between each individual observation and the mean. So that's xi minus x bar. And there are 100 observations, and many measures are close to the mean. And many are far from the mean. But here's the thing to remember, you're squaring them. Why? Well, because by definition, if you took the sum of the differences of each individual data point in the mean, they would add to 0, right? So you're squaring them so that they're all positive. And the variance is the average of the squares of the differences between each observation and the mean. So in this case, the square of the differences is 13. So one thing I want you to keep in mind is that a population, like all the dogs in the world might be your population, they will have an absolute variance in their running speed. However, we never measure every individual in the population. We measure samples. But you can calculate the variance from a sample. However, a finite sample is a finite sample is going to be a little less variable than the true population. And so we correct for smaller samples being less likely to actually catch the most extreme values with a little correction. We don't take exactly the average of the squares of the differences. We take a number that's a little bit bigger than the average, because instead of dividing by n, we divide by n minus 1. So I'm just going to say for samples, and I'm not going to be testing you on the difference between sample variance versus population variance, but it's like useful to hear because it didn't make sense to me a bunch of times. For samples, we divide by n minus 1, and it was a racist eugenicist who was a brilliant statistician who figured out that you can divide by n minus 1, and that'll be a big correction for really small samples, and it'll have almost no effect for really large samples, but really large samples are almost the same as the entire population. So you don't need a big correction for a large sample. So that's this minus 1 in the denominator ends up being this brilliant thing. Because small samples underestimate variance. So when I'm writing it here, it's not that I expect you to read my handwriting. I expect you to hear my words, and by my writing it down, that gives you enough time for you to write it down as well, right? So just like as a tip for how I think you should be taking notes in lecture. So the standard deviation is the square root of the variance. And that what that does is it gets it down to the scale of the actual numbers instead of squares, right? So you take the square root. So it's not the same as the average difference of each point. That would be nice. They should have done that. But it's kind of like the about how far on average are each point from the mean. Okay, so the important thing is not the math of how you measure variance. The important thing is what contributes to differences among individuals. And some of it are genetic effects, and not just all genetic effects, but especially additive genetic effects. So those are cases where having no alleles of a certain kind will give you one phenotype. Being homozygous for that, for a given allele will give you a different phenotype, and being heterozygous is halfway between the homozygotes. That's additive genetic variance. But there's other kinds of genetic variance, sources of genetic variance, like dominant and recessive genes, and also gene environment interactions among genes at different loci, epistasis. All of those are sources of variation, along with straight on regular environmental effects. Some of the dogs were well rested, and some of them live in a stressful environment. Some of the dogs have ad lib food, some of them have limited food. I don't even know what's better for running speed. Some of them got lots of exercise, and some of them don't get exercise. All of these are environmental effects, but a lot of them are also gene environmental effects. The dogs that are faster, um, the owners have more fun watching them run, or more likely to go for runs with them. The dogs that are slower, the owners won't take them for long walks and runs because they're constantly having to pull them along. A gene environment interaction. So all these things contribute to the variation among individuals. There are three complementary, but not exactly the same, definitions of heritability. The first is the additive genetic variance over total phenotypic variance. So VA, I'm going to type it so it's going to look nicer in the slide. And then we're going to make this divided by and so that total phenotypic variance includes wait 1 second, I wanted to see if I can, no I can't, includes sorry, I just want to make this bigger. It includes additive genetic variance, but it also includes variance due to the environment, variance due to epistasis, variance due to dominance, variance due to gene by environment interaction. So all of those go into phenotypic variance. So heritability is how much of all the differences among individuals, not how much of the trait, how much of the differences among individuals in that trait in a single environment are due to additive genetic variance, some of the differences due to genes. Another definition of heritability is the slope of the mid-parent offspring regression. So if there's a one-to-one correspondence, then heritability equals 1. If the trait is completely environmentally determined, all the differences among individuals were due to the environment in both the parents and the offspring, then who cares what the parent's phenotype is? The offspring could be large or small, fast or slow. This would be, sorry, I'll do a different color line. This would be heritability equals 0. So heritability always goes between 0 and 1. If there's no relation between offspring and their parents, the slope would be 0. If there's a perfect correlation, the slope would be 1. And then heritability, and this is the reason why we use this weird definition of heritability, that is not the same thing as genetic determination. Heritability will predict and can be derived from the response to selection. So the response to selection is the difference in phenotypes between parents and their offspring, and that equals heritability, which goes from 0 to 1, times the selection strength, the difference in phenotype in phenotype between the population before selection and those that reproduce. So we're going to do an example in the next few slides of what happens if we only allow the 10% fastest dogs to breed. We're going to do an artificial selection experiment. So actually, I didn't do the 10%. I did 50%. So the mean of this population is, is 12.7. Okay? So the selection, and the overall mean before was 10. So the selection differential equals 12.75 minus 10 equals 2.75. So what's the offspring mean going to be? Is it going to still be 10? That's the case if all of the differences among individuals is due to genes. Is it going to be 12.5? Sorry, 12.75. That would be the case if heritability equals 1. Or is it going to be somewhere in between? And the answer for a lot of traits is often somewhere in between. So let's say that heritability of this trait of running speed in dogs in this environment is 0.4. And the interesting thing about heritability, remember, is that it depends on the environment it's measured in. So if you took those dogs and moved them to a more variable environment, the heritability would actually go down, even if the genes are the same. That's worked out in the Ned Block article really well. Read it closely. So let's say the heritability is 0.4. In that case, the response to selection equals heritability times the selection differential. Selection differential, I already said, is 2.75. So the response, the change, which would mean the change from parents to puppies, is going to equal 0.4 times 2.75 is 1.1. So that means the next generation equals the original running speed plus 1.1 equals 11.1. So if you only let the dogs that can run on average 12.75 meters per second breed, some of the variation among the parental generation was due to the environment, some of it due to genes, 40% was due to genetic differences, added genetic variation. And so you're going to get a response to selection that's significant, but it's not going to be the same as the fast parents. So there you go. And in artificial selection regimes, this concept of heritability is incredibly powerful. You solve for the heritability, you will know how much your population is going to respond to selection. Of course, it's only going to last for a few generations because you're going to run out of that genetic variation that was in the original population that you did selection on. we saw that in the nest building in mice. So how is heritability measured? It's measured looking at correlations among relatives, doing those mid-parent offspring regression. But remember that the parents, the environment of the offspring and the environment of the parents are likely to be correlated. So if you want to do correlations, mid-parent offspring regression, you have to be able to randomly assign offspring to parents or adoptive parents, right? And we're going to talk in a second about how like you can't do that with people, not just for ethical reasons. The other way that heritability is measured is in selection experiments. We went over the example of the nest building in mice by Carol Lynch, so I'm not going to go over that example more because I already went into that in some detail. But we do measure heritabilities of traits in humans, in particular, heritability of IQ. This book in 1994, I think of it as a recent book because it came out while I was in grad school. But that doesn't mean it's recent for you guys. It was still long before you were born. Very influential book. The argument was, hey, we've done lots of heritability studies of IQ, and they're significant. The heritability in twin studies is often between 0.4 and 0.6, large heritable component within whites. And the average IQ of whites is about 15 points higher than that of African Americans, or at least it was in 1994. So some of that, if not all, of that 15-point difference must be genetic. So this is not a new argument with Herrnstein and Murray, but it was sort of notable that it was still being made in 1994, and it was very influential, and lots of people agreed with it. So Jensen made the same argument in 1969 that Richard Lewontin then argued against. And so when you read or have read the Ned Block article, that is, he does a really good job of describing the Lewontin thought experiment that we're gonna sort of talk about. So now I'm going to be talking about the Ned Block article. So Herrnstein-Murray's bias is partly like deriving from the start because they limit what's possible. In other words, Herrnstein and Murray say there's a 15-point difference in IQ, and some of that is going to be due to environment. You might think that most of that difference is due to the environment, or you might think that most of it is due to genes, but there's going to be some of it that's due to genetics. And what what Block points out is that Herrnstein and Murray limit they think that the environment can be at most 15 points. But let's say the effective environment on variation in IQ, or the effective environment, is more than 15 points. It's 30 points. And if you consider that, if you raise someone truly in isolation, they would not learn, language at all, the effective environment on IQ is probably much more than 15 points. And so if the effective environment on IQ is, much more than 15 points or 30 points, it's possible that African Americans are genetically 15-point IQ points smarter than whites, but there's such a huge environmental effect that it's swamping the genetic disadvantage that whites have, right? And that's actually not implausible, because as we're going to see soon, the effect of we actually know, we have great empirical data that there are large environmental effects on not just intelligence, which is hard to define and measure, but IQ, which is um, easy to define and measure, right? Um, so that was one point that Block is making. But then there's also the really fundamental point that the word genetic is somewhat ambiguous, and heritability is different than genetic determination. Okay? So we're going to sort of go through both of those. So as always, I talk about what's on the next slide, before turning the slide. So Bernstein and Murray um, define the range of possible um, causes of differences between groups in intelligence as there's this 15-point range. It's like what I said in the slide before. There's this 15-point range, and you could have extreme environmentalism, extreme determinism, and what they are then saying the reasonable view, which is that half of the differences in test scores are due to genetic differences that we're never going to fix. Um, However, there are lots of other alternatives, including that environment is having such a huge effect that it's overcoming a genetic disadvantage of being white. And because they don't describe that as a possibility, they're already biasing their views of the roles of genes in the environment. But then there's this other really critical point, which is that heritability is different than genetic determination. Heritability is a value that only has meaning and validity within a population and within the environment it's being tested. And it tells us nothing about differences among populations. Differences among populations could be due to genes, but you need a different kind of experiment, one that we can't carry out in humans, because humans create their own environment with their skin color. In other words, heritability, we're going to talk about this in a later slide, but I'm going to say it here. Heritability tests done on twins, done on adopted children, are only done within groups, within like racial groups, because it's understood that people carry their environments with them, because by looking different, we end up being treated different by other people. And that's what it is, but it also means that we don't have heritability measures that tell us anything about differences among groups. So this is the thought experiment that Lewontin talked about. Let's say you have a bag of corn, and it's a heritage breed of corn, so it's a little bit outbred and it's not all genetically uniform. It's it's somewhat diverse. And you plant that, you take a handful from that bag and you plant it in a seedling tray in a greenhouse with good lighting, like uniform lighting, everyone gets the same lighting, and the soil has high levels of nutrients. And when you do that, you're going to get differences among the heights of different plants, right? You're getting variability. And you then collect the seeds from those different plants and you rear them for another generation in that normal nutrient soil, and you'll get a measure of heritability, how much the differences among individuals the next generation are explained by differences among the parents, right? That's one definition of heritability. If you take a second handful of corn and you plant it in soil with low nutrients, you're still going to get differences among individual plants, right? And you can do a heritability study in that setting as well. And you'll get a measure of how much of the differences are due to differences among individuals. But that heritability measure, which will tell you a lot about like how much you could change the height of corn under high nutrients, and almost also how much you could under low, it tells you nothing about the difference in the means between the two populations. Heritability within um, populations or environments says nothing about the reasons, about the causes of variation among or between populations that are in different environments. Okay, it's a really important point. I think it's well explained in the Block article, and hopefully it's also coming through here. So there are kind of two senses of genetic. Genetic determination is the description of how genes act on traits during development. And a trait like, I mean, the example that that Block used in his article, finger number, is a trait that clearly is genetically determined. However, if you look in the population of people outside, there is some variation in finger number, and the vast majority of that variation is not due to genes. It's due to early experience, having your hand stuck. I slammed a door on my thumb when I was young, and so one of my left thumb is a little shorter than my right thumb. But I haven't had an accident of, fingers get chopped off in machines and using tools, or chemical insults during development can affect limb development and finger number. And so finger number is an example of a trait that is genetically determined with zero heritability. The differences among individuals are not due to differences among genes, but it is completely genetically determined, or mostly genetically determined. Heritability doesn't tell you how genes, whether a trait is determined by genes. It doesn't describe the effect of genes on a trait during development. It tells you not how much of the trait in an individual is caused by genes. It tells you how much of the differences among individuals within a single population that's in a single environment, variable, but a single environment, is due to genetic differences, additive genetic differences. So genetic determination is a property of individuals. Heritability is a population level trait. And I will just say, varies with environment. In other words, if you measure, you can measure heritability in a very variable environment. Same individuals will give you a lower measure of heritability than if you measured the same individuals in a uniform environment. And it's interesting, lab studies of heritabilities are often going to give higher values than parent-offspring regressions in the field, because outside is much more variable. The denominator of that heritability equation is going to be larger. There's a couple sort of if-then sort of relationships that Block explains. If a trait is genetically determined, heritabilities can be either low or high. If a trait is environmentally determined, heritabilities can also be low or high. And that's also because heritability can act through indirect interactions. So you might have a, a trait that is environmentally determined, like can you play the violin? No, if you don't have music lessons. Yes, if you have music lessons. But tiny differences in aptitude or ability can then lead to huge environmental effects. Like the parents see the kid doing, showing an interest in music, they then invest a huge amount in the environment, um, and then that child develops greatly as a musician. If they'd invested um, heavily in all their children's musical training, there would still be differences among them in their musical performance, but it would be much, 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 much less. Genetic determination doesn't really say how a trait can evolve, it shows how it can develop. Heritability will predict how a trait can evolve in a given environment. So they have different functions. And with IQ in particular, um, we have really good evidence that there's a huge environmental effect on IQ. It has a name, it's called the Flynn effect. And it was this observation that in many different countries, there were rapid changes in IQ scores recorded on a time scale of 1 or 2 generations, or sometimes less than a generation. So within a single generation, reference population 1962, usually IQ is always relative to a mean. So, within a cohort, the average IQ will always be 100, but then the scores get scaled based on different averages, and you can see how that scale changes, and that's what's going on here. By 1980, this I think might be in Denmark, it's going from 100 to 120. It was observed first in European countries. The Flynn effect is widespread. My graphs are not showing up, which saddens me. Let's see if I can copy and paste. I can't. That is so weird. Why is it not showing up? Um, I'm gonna share the an application so we can find it. Yeah, so I'm not sure why it wasn't showing up before, but this is in many different countries. Testing starts at different times. It's going to be starting earliest in England. Later, Israel doesn't exist as a country. You start testing here in 1991, but that same pattern is being repeated. Interestingly, it seems to be leveling off in some of the industrialized nations, not necessarily reflecting like degeneracy in the societies, but it's also possible that like these kinds of tests are something that um, you know cultures can get better at, and then once they've made those early gains, then there's sort of less improvement that's going to happen. But you can see changes over a period of decades of you know 10 to 25 points in IQ. Um, so there's an interesting, I mean, there's there's still this question, like we measure heritabilities within groups, within races, and they often seem pretty significant. Um, doesn't it mean something? Well, if we knew nothing except that there was a significant heritability within groups, then our sort of baseline starting assumption would probably reasonably be yes, some of the differences among groups is, Herrnstein and Murray are saying meaningful. What they actually mean is genetic. Um, I'm going to stop sharing the file, I mean the app, and I'm going to go back into um, the files here because then I can draw on them much more easily. So let's go back. Here we go. Great. Okay. Um, however, the fact that we actually know two really important things, one about the numerator, one about the denominator, means that we should not conclude that some of the differences among groups are due to genes, because we actually know, one, that there's a significant environmental effect on IQ test scores within groups, because we know it changes much more rapidly than genes can change, right? Whenever we have a drastic change in a trait that's taking one or two or three generations, think of it, as example, like obesity or diabetes. Um, we know that that change has happened far too rapidly to have been explained by genetic changes. There are genetic effects on obesity and diabetes, absolutely. Within groups, you expose them to the same, you know, horrible societal forces to get us to like drink soda and crap. Um, and some people will get diabetes under the in that environment, and some people won't. Um, but the fact that diabetes is increased on a scale, on the scale of um, tens of years, means that the increase in diabetes is not due to a change in genes, it's due to a change in environment. That doesn't mean that genes don't affect diabetes, but the change was caused by the environment. And so we have sort of the same data for IQ. We know that there's a large environmental effect, and then actually we also know stuff about. So that's actually in the denominator. We know that there are environmental effects. That's the denominator. And we also know that there's very little genetic variation among races, right? So that's the other thing. The numerator, we know stuff about the numerator as well. And so I'm actually going to skip the parts about reaction norms right now. Um, Although that's really interesting, and I'll probably have a chance to get back. I will get back to a little bit later. I'm going to skip to what we know about genetic variation among human races. And I know I've gone over time, and if you have to leave, that's okay, but I'm going to keep recording and I'm going to finish these slides. Um, okay, so um, interestingly, when evolutionary biologists talk about races, they are talking about something different than human races. Um, so when evolutionary biologists talk about races, someone like Ernst Mayr, they're talking about geographically distinct, usually with physical boundaries that limit dispersal, populations with different characters. Um, Races in sort of biology, when we're talking about non-human animals and plants, are basically on the level of what are called subspecies. There's significant genetic variation, limits to interbreeding, and they're probably in the process of becoming species. So that's usually easier to recognize for species that don't move that much and have a wide geographic range. But other examples are, like I talk about in intro bio, the apple maggot fly, Ragolitus pominella, which is a species that is used to specialize on crabapple, made the host shift to apple. Lots of interesting genetic variation, including imprinting on the hosts, that now there are subspecies with different timing of the overwintering period. And that's a genetic difference that's making them less and less likely to breed with each other. They're on the way to becoming species. Humans um, are not human races are not races in the Ernst Mayr sort of biological species concept sense. Um, there is genetic variation in humans, and we certainly look different, and some of us have highest heart disease under a high cholesterol diet, and some of us have the highest risk under a low cholesterol diet, although it's good for all of us to avoid cholesterol. Um, Humans vary on average by 1 nucleotide per 1,000 um, uh, base pairs. And um, some of that genetic variation among human populations is likely adaptive, although it's a small list of traits that we know about genetic adaptive variation, right? We know about the value of dark and light skin in terms of accumulating um, vitamin D and also avoiding um, um, and also trade-offs for avoiding skin cancer, but mostly the ability to accumulate enough light to produce enough vitamin D in short day environments. We also know about genetic adaptations for being able to tolerate or resist malaria, that in the heterozygote um, um, form, the sickle cell allele confers some resistance to malaria, which overcomes the downside of the homozygote having a debilitating disease. And probably one of the best examples, the ability to digest milk sugar as an adult, which has evolved multiple times in European and African populations, um, always associated with the cultural transition of raising cattle. And that's about it. There's an earwax gene that varies, but no one knows what the adaptive benefit of that is. And there's evidence of selective sweeps around a number of genes in the past. Some may be associated with severe plagues, but um, but that's about it. Um, One thing that's really interesting to remember or think about, which we didn't know when we were, you know, before being able to sequence genomes, is that um, the total amount of genetic variation among 7.8 billion humans is um, much less than is found among, um, I think, about 250,000 chimpanzees in the world. So this is a gene tree that is scaled to genetic difference. And so 7.8 billion humans, this is all the genetic difference that's observed compared to 250,000 chimpanzees. Um, and this is interesting, right? It didn't have to be that way. There were alternate hypotheses about how humans evolved when we migrated out of Africa, spread across the world, and which migration sort of led to the human populations that we see all over the world today. Um, and from the fossil record, initially there were thoughts maybe it was, the Neanderthals that gave rise to humans. And so it was a migration, that would have been a migration, like 200 or 300,000 years ago. Um, and it turns out that that isn't the answer. That the reason why we humans are so genetically homogeneous, which we didn't have to be. Wide-ranging species in the world are usually much more genetically diverse than humans, much more. That's because they spread across the world millions of years ago and diverged slowly over that whole time. As it turns out, what's also compatible with the archaeological record and DNA clocks is that a small population of humans of about n of 12, estimated to be effective population size of about 12,000 individuals, um, left, some of them left Africa about 52,000 years ago. And the combination of a population bottleneck and recent origin. So anatomically modern humans, totally interesting, hard thing to define, somewhere between 100 and 200,000 years old. But the amount of genetic variation explained today and the patterns of genetic variation are compatible with a very small population about 52,000 years ago. So it might have been a larger population, an older population, a more diverse population that was forced to go through a genetic bottleneck. And that's interesting. And that's not the way, that's not the only pattern of genetic variation that could have been possible for humans. It's just an interesting empirical fact. So this has sort of many repercussions. Um, um, the 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 greatest genetic variation among human populations is found within Africa. So there isn't an African race, and any differences that you're going to ascribe to races among races, if you're going to define them like Nicholas Wade in A Troublesome Inheritance based on continents, Asia, Europe, North America, Australia, and Africa, is actually um, those aren't valid sort of distinctions, because within Africa you have more genetic differentiation um, than you have among or within any other geographic area. But the other thing is, if you look on a finer scale, there are no distinct geographic boundaries or populations. Like in animal populations, you often find tight hybrid zones, usually maintained either by hybrid incompatibility or by like geography, like a mountain range that frogs have trouble crossing, or a river that mammals have trouble crossing. And then on each side, you have quite distinct, even different loci that are fixed for different genes. There are no fixed loci that are different among sort of continent defined races. And continents are probably a better definition of race than back when people defined races by countries, like Irish race and Italian race. Um, if continents are used to define humans, 85% of the genetic variation is within human populations, 10% is among, and those genes, those genetic differences are statistical. In other words, they're variation in the proportions of alleles. So there aren't unique alleles in different races. What you have is, at a given locus, you could be 40% type A blood, A blood type, A allele, and 30% B, and in another continent, it's 30% A and 60% B and 10% O, right? So the differences are in proportions of different alleles. Now, if you look at 10,000 different loci, you can be really precise and accurate in looking at a genome and saying the person that had this genome came from a certain geographic area. But that doesn't mean that there are fixed genetic differences between those geographic areas, which is really, really critical. And there are practical take homes for this as well. This is why you can have individuals from different continents that share more alleles with each other than, pair of individuals from a single population. And when there are only a few genomes that have been sequenced, we had some for some famous geneticists, Craig Ventnor and James Watson. It turns out share more alleles with Sungjin Kim, I think it was a Korean prime minister, than either do with each other. That having been said, James Watson managed to still think that people from different countries and races have differences in intelligence that are due to genes and not environment. So, no one is immune from typological thinking, which is a somewhat depressing take home. It also means that like some research programs that are being pursued quite actively might actually not be terribly productive. In other words, there's like racial pharmacology is this whole idea that we should be fine-tuning medications to different racial groups. And I mean, looking at the different effects of medications on genetic men and women probably is quite meaningful because there are a lot more genetic differences between men and women than there are between members of different races. And what it actually means is it doesn't mean that genotypes and medications don't interact. They absolutely do. But it means the focus has to be on specific genotypes, not on different racial groups, right? And I will end here and definitely read Ned Block's article really closely. It is extremely helpful. I'll post study questions for the heritability quiz, but I'll do that tomorrow because we're going to do the regular quiz tomorrow. And I hope you're interested enough to go into Blackboard and look up some of the other articles that I posted because it's interesting. Okay, if there are any questions, next week there's no class. Good point. So you'll have a lot of time to study for heritability, but only short time study more for tomorrow's quiz. Good point. Yes, quiz will be in lecture. The heritability quiz will be in lecture on the 23rd, but I'm going to open up the quiz that I was hoping to give today tomorrow at 12 noon.